the title.
Yeah, good afternoon all. Thank you for an opportunity to present our recent research in collaboration with Bohdan Lev, Miha Ravnik, and me, Oleksandr Chernyak. We are here to... Okay. Maybe. Yeah, we are here to present some insights on possible bacteria motion mechanism that are quite novel. So I will build my talk as follows. We discuss some motivation, then we give some properties of our main suspect, Chromatium oceanibacteria. We briefly remind some bacteria self-locomotion mechanisms. Then we discuss what we propose and the results that we obtain. So, we know that we usually see a lot of aqueous environment, and in this environment there are some processes like in the lakes that can distinguishingly classify these lakes in two separate classes. These lakes can be classified by such process as stratification. It's the tendency that lakes can or cannot have some distinct thermal layers within them, and typically there are three layers. If they have, it's top warm layer, it's some intermediate layer that can change its volume during the day, and some bottom cold layer that are not particularly interesting for us, but this classification is important since there are two types of layers: those who intermixes and those which don't. The layers which don't intermixes through the whole volume, they are very widespread, but the lakes that don't intermix, they less widespread, but have very vivid characteristic like their color. The ones which don't intermix, called meromictic lakes, and as you can see, especially at this slide, they have some colored water which are not usually seen in rivers or oceans or whatever. So the reason why this color observed is actually in their inhabitants in this top warm layer of lake. And as you can see here, it's bacterial layer that changes the color through refraction or reflection or whatever. We are particularly interested in Chromatium oceanibacteria. It belongs to some class of purple sulfur bacteria, and the main characteristics possess that it in water inhabits water, it capable of photosynthesis, it has a form of straight or lossy curved rods, and it is single-celled bacteria with length about 10 micrometers. It fully filled with globulus in range of 1 to 3 micrometers, and it can have or it can have not flagella. The main interest of previous research was the color of this bacteria. It can be purple or reddish, but recently in Switzerland, there were found a lake with this bacteria which has green color, and people tried to explain why is that. During these experiments, it was observed also that bacteria without flagella can also move. And during this motion, it changes its shape, interface of bacteria. So that arises another interesting problem of explanation why this bacteria moves and what mechanism can explain this. So typical bacterial motion mechanism are flagellum, which rotates and pulls or pushes bacteria, fimbra, which are a lot of hairy-like appendages which can crawl along some surface, or gliding, which less understood and less explained, and for some cases like Myxococcus, it can be explained through the focal adhesion complexes, but it's not like in every case that can be used. So we know from previous research that bacteria can deform its interface during the motion. So we would like to use that. And in the work by Sell and Endelman, there were formulated an approach using free energy to explain the pattern formation in two-dimensional or three-dimensional materials. And we will build our approach atop of two main statements. First, that we observe deformation of bacterial interface. And second, that this deformation with interaction with media gives a motion to our bacteria. This free energy can be split in two parts. The one which describes the dipole interaction, and the other which describes the curvature deformation of bacteria interface, which also caused by dipoles inside the bacteria. In this research here, we propose some solution or equation, at least, for curvature deformation according to the given polarization at the surface of bacteria for plane geometry. But obviously we have a rod, and this is just an example that we in fact can have the deformation of this interface, but in real case we need a little bit more study to correctly describe all the process. So what we suppose, to be precise, we have a bacteria in a layer in waterous media that are enlightened by, well, natural sun or other lightning, and this light causes the polarization of globules inside the bacteria. They polarized, and also by this light caused thermodiffusion, which moves them inside the bacteria. They have some non-uniform distribution, And this non-uniform distribution at the interface of the bacteria causes the appearance of this deformation, which are moves because it's a non-uniform distribution or just a deformation, and it can be like soliton, and thus it evolves along this bacterial interface. This, if we consider only deformation, that can be explained as it can be seen as a turbine, or if we consider only polarization, that can be considered as a polarization current interaction with media of random electric field caused by ions and in water. So we try to describe this mechanism by, again, using free energy. We describe here only polarization now. So this is the typical free energy with existence of gradient of polarization. We expect or suppose that we have no charges on the bacteria interface. We expect that we have a rotation, vortex, vortex at this interface, so that this free energy can be rewritten as follows. And by minimizing this free energy, we have the equation which describes exactly the polarization. But it should be noted that this polarization is only along the bacteria interface at the interface itself, and we exclude here rotation by just this solution. So that can be written as follows, where phi is polarization, dot phi is gradient of polarization, and the coefficients C beta, C kappa, and K alpha is just the corresponding coefficients beta, kappa, and K divided by alpha. So, So this equation is difficult to solve. We considered 3 cases, but it's like kappa 0, where we have no perturbations. Small perturbations was considered as a perturbation theory, and when we have large perturbations that can correspond to some interesting situation with bacteria splitting, we have the last equation, and here we present some typical profile that can be seen as a solution of these equations. That's in fact, that's a number of such curves. It's periodic. In fact, this function is 2 periodic, but we're interested only in real values. And according to some constant like Cphi and L, we can change this curve. The main needs is to be that this curve gives an zero polarization to all volume of the bacteria. And the next slide is a case when we have a large or extremely large perturbation that corresponds to the, we think, we suppose that corresponds to the case when bacteria splits. And by this, we see that the polarization is situated mainly at the edges, and therefore they turn apart these bacteria. And again, by using these initial parameters for this solution, we can see what mechanism will be, what will be. So another important task is obviously to describe the possible velocity, estimate at least velocity of such a motion. And for this case, we can switch to hydrodynamics description of soft active matter, and it is these equations are in fact presented in work by Marchetti et al. And that's just we need this only briefly, but this is just concentration. This is polar vector evolution of polar particles. This is continuity equation, this is dynamic equation, this is Gaussian noise, this is free energy, which we actually used above. And through this equation, we can estimate our velocity as proportional to variation of free energy, which we take proportional to the Frank constant because of previous solutions, and rotational viscosity, the length of bacteria, and it appears to be like 1/10 of micrometers. That can be seen as a good result because it is in the range with realistic values by magnitude. So as a summary, we estimated, we propose a novel mechanism of bacteria motion, and it is based on the idea of non-uniform polarization interaction with environmental random electric field. For example, we investigated some cases of small and large perturbations and seen the possible just possible motion or splitting of the bacteria, and we estimate velocity for such a motion, and it gives some realistic values by magnitude. Thank you.
Thank you very much for interesting material. And who wants to ask the question?
My question is,
No.
where in your equations the difference between life and death bacteria exist?
Alive and death?
Yes, your equations, difference between alive and death bacteria. Specificity.
Yes. I just okay. I can move only one direction. It doesn't. Can you? Okay. Well, it is much more correct, maybe, to say that it's not like dead or alive, but it's active or non-active. And active is described by self-propelled. Well, then I can. And active is just described by some self-propelled velocity, like this one, and that's all. If you have some other characteristics, like it's not alive, but some active particle caused by
Who can differ? Who can differ the alive from died? Maybe in parameters.
The dead doesn't move. Dead don't move. The dead ones. And alive or alive-like, they just move, and we have parameter of such motion, or we describe it in some way through the interaction with media.
Okay, please. Okay.
Is there any kind of bacteria with the jet mechanism of motion?
Jet, yeah, I heard something like this, yet I'm not sure what are exactly they are, so I cannot give an example, any example.
Next question.
Sasha, thank you very much. That's really interesting talk. My naive question, do these bacteria move in the night?
Oh, that's a good question. Actually, I don't know. I think they, the ones with flagella, I think they move. The ones with no flagella, I think no. But then again, there is always light, almost always light, except very, very, well, black box. So I'm not sure.
And another brief question. Do you think that your beautiful idea of the light-induced polarization can explain also our high level of mood activity when it is sunny outside and when it is rainy?
I'm not sure that it's directly, it's more like, maybe we have some, not a good word, parasites that are active or inactive during the light, and that correlates with our mood much more.
And my question is why you use interface, not surface?
It's just a common term in terminology in this science, as far as I know, because through this surface it can interact with media, so it's like some interface which you give input and obtain output.
Question more? So we finish our. Thank you.